Hello everyone. Today's demonstration is tissue processing of soft tissues by paraffin embedding technique. The first step is obtaining a specimen. The specimen is obtained in a clean jar containing 10% neutral formalin. Once received, the tissue has to be cleared for all the debris and blood clots, and it has to be placed in jar containing water for running water for 24 hours. The tissue is kept in water so that the formalin is washed out. Then the tissue is taken in a petri dish for labeling. Labeling of the tissue will help to maintain proper records. Labeling is done by an alphabet B, then a number denoting a tissue number and the year. The next step is placing the tissue in a cassette. The cassettes are different for an automatic tissue processor and for the manual tissue processing. The plastic cassette is for automatic tissue processor and a steel cassette is for manual tissue processing. The tissue is now taken for the grossing. For the grossing, the tissue is first measured by the scale to get its gross details, and then a cut is given with the scalpel blade. The cut section will help us to direct the tissue for the orientation. The tissue is now placed into the cassette. After grossing, the tissue is placed into the cassette. The cassette is packed and the processing begins. The first step of processing is putting the cassette into the formalin for the fixative. This will fix the tissue, will coagulate the proteins and prevent the tissue from autolysis. The tissue is kept into formalin for around 24 hours or depending upon the density of the tissue. The time may vary. Once the tissue is fixed, now it has to be dehydrated. The dehydration is done by placing the tissue into the various grades of alcohol. To begin with, you can start in 40% alcohol, 60% alcohol, 70% alcohol, 80% alcohol. In each jar, the tissue is kept for approximately 3 hours. The time required is minimum 3 hours. Lastly, the tissue is put into an absolute alcohol. 2 changes of absolute alcohol are given so that the complete dehydration of the tissue is done and the tissue is now ready to be cleared. The clearing is done by a clearing agent, that is xylene. Xylene is a solution which will remove alcohol completely from the tissue and make the tissue ready to be embedded for the paraffin. 2 changes of xylene are done approximately of 2 hours each or depending upon the density and the size of the tissue. Once the clearing is done, now the tissue is ready to be embedded into paraffin wax. For that we have an instrument known as wax bath. This wax bath keeps the paraffin wax at the temperature approximately 60 degree in a molten form. The tissue is kept into the paraffin bath for around 2 hours. Two changes of paraffin bath are done so that the clearing agent xylene is completely removed and now the tissue is completely embedded with paraffin wax. This tissue is now ready for embedding. The embedding is done in L-shaped Leucard's wax block maker. This L-shaped Leucard's blocker are for manual processing and a tray is for automatic processing. The tissue is now placed with the cut end. The tissue has to be removed from wax and it is placed with the cut end facing downward and wax is poured over it. It has to be removed from the cassette and placed into the mold. The cassettes for automatic is plastic cassette. Now the tissue is placed with its cut end facing downwards and molten wax is poured over it and it is allowed to set with its labeling sticker on the upper side for the identification of the tissue. Once set, the block is ready in this way. This is for manual cutting and for the automatic cutting, automatic tissue processor, we have such type of block. This is the way the block is ready for the further step, that is sectioning. Sectioning is done on the microtome. There are two types of microtome, manual and automatic. Here we are showing you on the semi-automatic microtome where we section the tissue. The thickness is set to 6 micrometer. The block is attached to the block holder. It is fixed tightly so it should not be slipped off. The position is set to bring it to the cutting knife edge and it has to be fixed properly so that the cutting surface is towards the knife edge and manually it is rotated so that you get a clear ribbon. You get a ribbon of wax containing the thin sections of the tissue. You have to take quite a big ribbon. First few part has to be discarded and then a neat continuous ribbon along with the section of the tissue is ready to be taken on the slide. The slide is coated with egg albumin. It's ready, already coated slide so that the tissue gets fixed to it. Now it is placed in a hot water bath so that the wax melts out and only a thin section of the tissue is taken on the slide. The tissue has to be taken without the folds. See, this is the way the tissue is on the slide along with the wax. The slide has to be numbered again for the identification. The same number what was given on the block has to be written with a glass marker or with a unerasable pencil on the slide. Now the slide has to be placed on a hot plate to remove the excess wax, and the tissue gets fixed on the slide. This slide is now ready for the further staining, that is hematoxylin and eosine staining technique. That will be our second part of the demonstration. Thank you. Here it's a chart showing you all the steps of tissue processing. Kindly go through it and revise each steps properly.